Log on, tune in, find out. Another good idea from Cambridge.
The last part of the afternoon session, which consists of one speaker. Before I hand over the word to James McInerney from Dublin National University,
Menus
University, I want to draw your attention to this offer by Cambridge University Press. There's 25% discount on all books for delegates of this workshop. I left the flyer out there, so just go and grab them if you're interested. That's what I wanted to say. James, please.
Okay, thanks very much.
Is that okay? Right. So first of all, I'd like to say a big thanks to Vince and Kathy and all the organisers for inviting me here. Whoever invited me, I got the email from Vince. But I'd like to say thanks very much for them. I think it's a really, really nice meeting, and the thing that interests me most was the fact that it's a very non-tree-like meeting in the sense that there's a lot of horizontal transfer and hybridization, all of these non-tree-like processes being talked about this week. I'm organising next year, for good or ill, SMB 2012, so we have 1,300 people coming to Dublin next year to talk about different things in evolution, I guess. And I thought maybe, I'd first of all invite everybody to come if they'd like to come along, and perhaps even to propose a symposium. We're going to have 24 symposia, so if you guys thought that it was interesting to continue in this theme, certainly in June of next year in Dublin, you're more than welcome to do this. I'm going to talk about two different things. First of all, Lars yesterday talked about something very, very similar to the first part of my talk, so I thought rather than sort of labour for a full hour talking about non-tree-like methods to look at alignments, I would talk about that in the first instance, but at the end perhaps we'd talk maybe a little bit of philosophy of science. I'm working with some excellent philosophers, one of whom is Eric Baptiste, who does some great work on non-tree-like thinking, and I thought it might be interesting to talk about that here. So overall, I guess the theme that pulls this together is that we might think outside the tree. So first of all, if we talk about multiple sequence alignments, and if we talk about trying to make good phylogenetic trees, there's a very, very good quote from Darwin where he said the importance for classification of trifling characters mainly depends on their being correlated with others of more or less importance. In other words, character correlation is a very, very good indicator that you're in some way classifying the organisms that you're looking at, that you're classifying them well. You're doing a good job. So this idea has been around obviously for quite a long time, and people have worked on it for quite a long time. And a very interesting and significant paper was published in June 1969 by Walter LeCain. And Walter LeCain worked from home, and this paper here is a method of selection of characters in numerical taxonomy. And what he introduced at that time, there's a little bit of, I'm very, very reluctant always to talk about history, because you can say, well, this person introduced it, and always somebody's going to come along and say, no, it was invented 2 years earlier by somebody else. But Walter LeCain's paper is particularly important because he describes very clearly character compatibility and how you might decide that some characters are probably very, very good for phylogenetic analysis, but other characters might not have that sort of property that they are going to be good for resolving all of the trees. And what he did was he looked at characters, pairs of characters, and he asked the question, can we fit both of these characters onto the same tree? Is there a tree that exists onto which we can fit both of these characters? So two characters can be said to be compatible, and so this is the beginning of compatibility analysis, if they can be mapped onto the same tree without homoplasy. And this is table 2 from his diagram, where he's looking at Argodrepana, which are a group of lepidopterans, and he's got a variety of morphological characters that he's looking at. And in a pairwise fashion, he analyzes these characters and he puts an X into the cell if you've got two characters that can't be fit onto the same tree. And he says these two characters are compatible. And then by taking a sort of broad look at the data, he said, well, I guess this character here seems to be pretty much incompatible with lots and lots of other characters. And these characters seem to be incompatible, and over here we've got some characters that are incompatible with lots and lots of others. And so from this he sort of says, well, these characters in total seem to be not very good or not very useful for phylogenetic analysis, The other characters are compatible with each other, so maybe we'll go look at those kinds of characters. So that idea has been developed down through the years by people like Chris Meacham, and Mark Wilkinson has been working on this, and there's lots and lots and lots of people have worked on it. And Davide Pisani, who works with me, he published a paper in 2004 called it's Identifying and Removing Fast Evolving Sites Using Compatibility Analysis, an example from the Arthropoda. And so, having spoken to Davide about this kind of method and about compatibility analysis, it sort of occurred to me that perhaps characters would still remain useful, even though in total these characters can't be mapped onto the same tree without homoplasy. Is there some way we can sort of allow a little homoplasy? And maybe that's still going to be a useful thing. Davide worked on something called Lekain probabilities, which is, as I said, derived from the 1969 paper that Walter Lekain wrote. He defines the probability of a random character having as low or lower incompatibility with the rest of the data than does the original character. So Davide looked for characters that really contributed nothing more than randomness to the data set and said, well, we might safely remove these kinds of things. So that's one kind of approach. It's a compatibility kind of approach, and it's on this kind of approach that I've sort of built what I'm presenting here today. But there are other methods for dealing with not so nice characters in evolutionary biology, not so nice characters in alignment. And in December of the same year, 1969, another very important paper was published. This was published by Steve Farris, and he called this a successive approximations approach to character weighting, or it's quite often referred to now as SACW, successive approximations character weighting. And so the idea is that I guess initially most characters get an equal weight. We consider them all to be equally good when we're inferring phylogenetic trees, and Farris proposed that maybe we might use successive rounds of weighting in order to downweight some characters or maybe upweight others. You can weight them different ways. The method principally involves inferring a phylogenetic tree, an initial phylogenetic tree, and based on that phylogenetic tree, looking at the consistency of the data with the tree. So he described consistency indices in this paper. And a consistency index is 1 if a character can be fitted onto the tree without homoplasy, and it's less than 1 if you have to invoke some kind of ad hoc hypotheses or some homoplasy. So this was another way of discriminating between characters in an alignment. And I guess the most popular, most commonly used method now is where we try to model different characters in alignment, model the process, say that constraints on evolutionary change are different at different parts of the alignment. Some parts of the alignment might be rapidly evolving, some might be slowly evolving. We try to model this, and the most widely used model is the gamma distribution. And generally we use a discrete approximation to the gamma distribution, and I don't have to tell you guys about this because you know far better than I do. So this is a selection of methods, a non-exhaustive selection of methods. Compatibility, successive approximations, character weighting. I didn't talk about slow fast, but this comes from Henner Brinkman and Hervé Philippe, and it's a method again of discriminating between characters. Maximum likelihood assignment of rate categories to sites. So again, this is kind of, if you like, an update of successive approximations character weighting, but applied to maximum likelihood, where you infer a tree, and then using maximum likelihood, you estimate the evolutionary rate of each site, and then you can put them into categories or into bins, and you can include or exclude characters according to that. And it's quite popular, and I've done this myself in the past. Another one is just to evaluate the number of character states, and to use the number of character states at a particular position, at a particular site, and say that the number of character states is a proxy for evolutionary rate. The more character states you see, the faster the evolution of that site. If you see no variation in the site, that's a slow site. If you see lots of variation, that's a fast site. Many of these methods are tree-based, and some are not tree-based. So pairwise comparisons of sites are not tree-based. Just even counting the number of character states at each site, that's not tree-based. Pairwise compatibility, that's not tree-based. Some of these are tree-based. And what I'm going to show here is the problems that exist if you use a tree-based method for trying to evaluate the rate of change at a site. And I'll tell you the answer before I go through it. If you use a tree that's not right in the beginning, then you may get an incorrect rate assigned to a particular site. If you do use the correct tree in the beginning, well, what do you care about the rates of the sites? You've got the correct tree. It's fine. Don't worry about it. So there's a bit of an issue. This is just a very simple cartoon. Here we've got a particular site. We've sampled taxon 1. It's got an A nucleotide. 2 has an A nucleotide 3. So on this particular tree, if we presume that this is the tree, then this is a relatively slowly evolving character. However, if we find that this is our best initial guess at a tree, then this exact same character is now rapidly evolving. So everything depends on the first tree. And I'll go through this. People have recognized this for a little while and have tried to find ways around it. And probably the most innovative way I've seen is from this paper by Fisher and Palmer in 2005. 2005, yes. And what they did was, very, very simply, they took this core tree that was fairly uncontroversial. And they have some controversial lineages that they want to find the positions of those lineages on that tree. And so they produce a whole group of trees. And from this group of trees, they go through a variety of different, I guess, manipulations of the data until finally they have a stable topology. They've evaluated which sites are rapidly evolving, which are slow evolving. They've got this iterative method here that's not terribly different to successive approximations, character weighting, but effectively they're not using a single initial tree, they're using many initial trees. It's quite good, but at the same time, we still depend on knowing some of the phylogeny and knowing it with certainty. If we're going to try and sort of evaluate characters and character rates, then there's a couple of things I think that are probably desirable in any of these methods, or desirable outcomes in some cases. One is to improve the bootstrap support. You might like to improve confidence in your phylogeny. However, the issue is here. What do bootstraps mean? If you take a gene that you've sampled, you've sequenced the gene, you've aligned the sites, and now you manipulate the alignment, getting rid of the things that you don't like in that alignment. And then you see high bootstrap support. What does that mean? I think it's okay to do that, but I think you need to be very clear in what you're doing when you're doing it. And you need to be very clear on how you interpret that. And a bootstrap support in that case might be something like the probability of observing an internal branch in a tree, given the particular analysis method, provided, the probability that you would see that branch, given a particular analysis method, if you were able to sample all of the data, or the whole universe of data, but no, you're disagreeing with me completely. I can tell. Anyway, we could talk about it, but you have to...
I just want to know, do you mean bootstrap support for the individual edge or of the whole tree?
Bootstrap support for individual edges, yeah. I think there are issues there. I think you can interpret it, but you need to be clear on what it is. And I think it doesn't mean the same thing as the original bootstrap interpretation does. But I think you can explain it. Improve reconstruction of deep branches. That's something that's problematic. I think everybody agrees there's an issue there. Deep trees are difficult. Reduce things like composition bias, and improve the chances of recovering the correct tree. So those are kind of desirable things. So the way in which we look at it is to take a look at each single site in an alignment, but also this can work for morphological data. It's just homologous characters. It doesn't matter what they are. I'm going to mostly look at amino acids and nucleotides, but it's perfectly acceptable to do this using morphological data or any other kind of evolutionary data. So you take a particular character, and the distribution of character states forms a set. And we can partition this set according to the character states. So this is the set of character states, and this is a partition of that set according to the nucleotides that are here. So 0 and 1, taxon 0 and taxon 1, they go into here, they form a group. 2 and 5 are here, and 3 and 4 are here. So the nice thing about this is that when you do this kind of little transformation, it doesn't matter what the nucleotides are. If you have an identical distribution, but instead of A's you see G's, instead of T's you see A's, and so on, then this particular coding means that those two things are comparable, or are much more easily comparable. You can do it for nucleotide, as I said, you can do it for morphological data. So what we're hoping for is, in some way, to have a proxy for evolutionary rate. We could evaluate the actual evolution of a character if you knew the correct tree and you had the model of evolution or the process of evolution available to you that was correct. So those are difficult things to come up with, but we may have a proxy. And the proxy that we have here is R being the rate at a particular site, summing over all pairwise comparisons of sites or of characters in the alignment. We look at the partition agreement. In other words, the similarity of two partitions to each other, two partitions of these two characters, these two sets. So we summarize this, we find what that looks like when you compare a site to all the other sites. Okay, the partition agreement between site I and site J is this here. This is going to be published in Systematic Biology, so you can go have a look at it. But it's always nicer to have a little cartoon, I think. So if you've got an alignment like this, you can take a particular site. Let's say this is the distribution of characters. Let's say here's another site. That's the distribution of characters. You can recode them. So 0 and 3 both are grouped together. 1 and 2 are grouped together. 1, 2, and 4 are grouped together here. And over here, 0 and 3, 1, 4, and 2. And so it's not a symmetric distance between them, the distance that we've used. We did evaluate about 5 or 6 different distances, and this is the one that we find works best. So it's just a method for data exploration, but in our hands, this is the method that works best. So when you compare this site with this site, we get a distance of 0.5. When you compare this site backwards with this site, you get a distance of 0.33. Okay? Groups in this particular set are said to be compatible with these if they're the same as them or if they nest within them and the same backward. Okay, so you can get just a pairwise comparison. You do this if you've got 1,000 sites in your alignment, you compare this with the other 999 and so on. And so in the very same way that Walter Lecain had all of the pairwise comparisons between the characters in his matrix, we can carry out this. It's okay, there's software for it. And so we had a look then to see, does it improve things? Does it do something? The idea being that if a character is rapidly evolving, then the distribution of character states is going to be weird. It's going to be very unusual for the data set. And the true evolutionary characters will tend to have particular kinds of patterns. And highly homoplastic characters will not tend to have those patterns. And that's what we're trying to figure out. So, as usual, we have some simulated data sets and we have some empirical data sets. I don't have anything like a proof, but I think it's just a work in progress right now. So, if you've ever bought Maclade or downloaded Mesquite, there's a beetle data set, 49 taxa. It's a nice real data set, a little bit balanced, a little bit imbalanced. So we use that data set because it exists in the literature, and we made an alignment, 999 base pairs. Using SeqGen, we produced the alignment, and we put it through the software, and we put all the resulting characters into bins, in this case 20 bins. The fastest evolving characters, according to that RI, are here. The most slowly evolving ones are here. We did 2 simulations. One, modeling among site rate variation with a gamma distribution set where alpha is set to 20, and with 4 categories of sites within that simulation. The other one, the gamma distribution was set to 0.5. What you can see here is that I guess the 2 simulations, and so our method, which we call Tiger, tree independent generation of evolutionary rates. Tiger picked up 2 different patterns. This one looks a little bit L-shaped. This looks a little bit hill-shaped. Might be what we expect. The interesting thing is here, that when you simulate the evolution of these alignments using SeqGen, using 4 discrete categories approximating the gamma distribution, what we found was that there was 4 groups of sites coming out. And it simulates them according to the average rate in each category. And in fact, rather than seeing a smooth L shape here, we see 1, 2, 3, 4 different categories. So it's picking out a very, very subtle pattern in the data here. So that's the first one. How about deep branching trees? Does it do anything here? So this is a difficult phylogeny to resolve. We have a deep branching tree, fairly long branches. These ones quite long, very short internal branches in places. using maximum likelihood or maximum parsimony, it's very, very difficult to recover the tree that we use to simulate the data. If we lop off the most rapidly evolving sites, according to this tiger method that I'm talking about, then we get resolution of some parts of the tree. Not the more difficult ones, but parts of the tree came back. We removed the sites that seemed to be the most confusing for the phylogenetic reconstruction methods, and we got some resolution in the tree. So that's the kind of thing that you're hopeful about. So our next one is we used 8 taxa. So we used 8 because we can look at all 10,000 odd trees for 8 taxa. And we can ask what's happening to each one of those trees as we remove the sites that we think are most rapidly evolving. So it's a very, very simple and quick analysis. In red is the consistency index, which is what Faris described in 1969. Again, a consistency index for a character is 1. If it fits onto the tree without homoplasy, it's less than 1 otherwise. And you could have a consistency index being the average of all the consistency indices for an alignment. And so what we see here when we include all sites in this 8 taxon tree, we made 999 base pair alignment. The consistency index is a little over 0.8 for the true tree. We know the true tree because we made the alignment on this true tree. So if we pick 100 random trees and we ask what's the consistency index, it's somewhere lower than it is for the true tree, as it should be. When we exclude the bin 10, which is the category of sites that are the most rapidly evolving, then we see that the true tree, its consistency index improves, whereas the other trees that we randomly picked doesn't seem to improve. And the same happens here, but I guess the standard deviation is moving a little bit here. So a little bit more detail on this, it's probably a better analysis of what's happening, is this. Again, the 8 taxon tree, each dot here represents a tree. This is the similarity to the true tree, and so here, of course, is the true tree, the tree we use to simulate the data. This is just some measure then of the distance in topology between this particular tree and the true tree. And the method we used was something that's implemented in top-D FMTS. And this is Faris's consistency index. So for the unperturbed, unmodified data, the true tree gets the highest consistency index, as you would expect. But what happens if we start taking away the sites that disagree most with the rest of the alignment? Okay, what we think are probably the most rapidly evolving. So what we have here again is distance. The true tree is over here on this y-axis, and the arrows here are pointing to 0, which is no improvement, no change, no improvement or disimprovement in the consistency index when we remove bin 10, 9 and 10, 8, 9 and 10, 7, 8, 9 and 10. The tree whose consistency index improves the most is the true tree. Now remember, the Tiger algorithm didn't know what the true tree was. It was never given the true tree. But the consistency index that is improved by removing this particular category of sites, the consistency index that's improved the most is the one that is for the tree that was used to simulate the data. Removing two bins worth of sites, if that's how to say it. The true tree improves the most. Of the 10,000 trees, this is the one that improves the most. But what's happening to the rest of the trees? Many, about 2/3 of them have now a higher consistency index. They're more consistent. The tree is more consistent with the data as we remove these more rapidly evolving sites. About 2/3 of them improve, but some get worse. And in general, the trees that are most dissimilar, the ones that have most branches wrong, they're getting worse. So the method is discriminating against bad trees and in favor of good trees. Here, again, out of the 10,000 trees, the true tree is the one that improves the most, and here, out of the 10,000 trees, these start to cluster, and I think that's probably because of different branches that they're getting right. So these ones all have a particular important branch right, and so on. Okay, the Thermus data set, fairly well-known ribosomal RNA data set. There's 5 taxa in this data set. 3 of them are thermophiles. So it's Thermus aquaticus makes Taq polymerase. There's Thermatoga, and there's Aquifex. So 3 thermophiles and 2 mesophiles, Deinococcus radiodurans, which is a radiation-resistant bacterium, and Bacillus subtilis, both mesophiles. The difference between thermophiles and mesophiles generally, when you look at ribosomal RNA sequences, is that thermophiles have high GC content in their ribosomal RNAs, where mesophiles have a more moderate GC content in their ribosomal RNAs. This presents a problem for phylogeny reconstruction because of compositional attraction, the thermophiles tend to group together and the mesophiles group together. So you've got a hot part of the tree and you've got a cool part of the tree. Sorry. Okay, so just to let you know, we think this is the true tree. There are many, many other characters that unite Deinococcus and Thermus, menaquinones, flagella, this kind of thing. And we think this is the true tree. And indeed, if you use a clever method of phylogeny reconstruction like log det, then you can get the true tree. But let's just pretend we're a little bit stupid. And instead of actually developing a cleverer method, we're trying to manipulate the data in order to see what trends are in the data. So here I'm just going to compare a couple of different tree-based methods with the tiger method, which is not tree-based at all. So if you have no site removal and you try to make a phylogenetic tree using these, this is a thermus, which is a thermophile, thermatoga is a thermophile, aquifex is a thermophile. These are high GC sequences. So you've got the hot part of the tree and you've got the cool part of the tree. It's the wrong tree. We think it's the wrong phylogenetic tree. And you get not terribly good, but moderate support for that wrong tree if you include all of the data. Let's just suppose then we use maximum likelihood and we use tree puzzle as the tool for doing this. And we use 8 categories of sites and we remove the fastest category of sites. So you have an initial phylogenetic tree, which happens to be this tree. Okay? So regular maximum likelihood methods will recover this tree. So you have that tree, you remove category 8, the most rapidly evolving category, and now you see what your phylogeny is. And the only thing that's changed is you've gone from moderate support to absolutely excellent support, but it's for the wrong tree. Using successive approximations character weighting in a parsimony framework, same thing. So the initial tree is wrong, and so therefore you get incredibly high support for what turns out to be the wrong tree. If you use this tiger method, pairwise compatibility, no tree initially. It doesn't know anything about the trees. You give it no tree, it just looks at pairwise compatibility. Remove bin 10, the most rapidly evolving sites, and we finally get a tree where Thermus and Deinococcus are placed together. This is what we think is the correct phylogeny. And the bootstrap support, whatever that means, is moderate for these. There's another data set, a mitochondrial data set from the 1980s that's quite famous, and it looks at a bunch of apes. And this is an interesting data set as well for one particular reason. If you use maximum parsimony to analyze this data set, you get 2 equally good trees. They require 1,153 steps, and the 2 trees conflict. One places humans and chimps together, and the other places chimps and gorillas together. And if you bootstrap this, you get approximately 50% support for this topology and something like 50% support for that. So something like parsimony isn't making up its mind about these data. The thing about this is that likelihood does make up its mind. Likelihood prefers this tree here. But again, we're just talking about looking for patterns in data. So the primate data set here, you've got 2 strongly competing hypotheses for the raw, undisturbed data. We're going to see if the tiger approach has helped and how tree-based methods work out as well. So first of all, successive approximations character weighting. So we look at the initial tree, we can't decide. So how about using both trees? Let's look at successive approximations character weighting when we've got the human chimp tree as your initial starting tree. And let's look at it when you've got the chimp gorilla tree as your initial starting tree. So 2 different analyses. So if we use the human chimp tree as the starting tree, bootstrap support is 79% for the human chimp tree after you manipulate it. If you use the chimp gorilla starting tree, then you get 77% support for the chimp gorilla tree. So the starting tree decides effectively which tree is going to be more strongly supported the next time around. I mean, it does make sense. You have a tree, you're going to get rid of all the characters that don't agree with it, and then you ask the question, do I get a better phylogeny? We certainly get higher bootstrap support for that. Maximum likelihood site removal always recovers what we think now is the correct tree, humans and chimp, though with higher support for human chimp. So if I did the same analysis, with higher support when human and chimp is used as the starting tree than when chimp gorilla, that's a hippo, was used as the starting tree. And it removes different sites. And indeed, it's not that this is a subset of 121. There are some sites that are unique to each different thing. So the tree does have quite an effect on this. Tiger site removal, we removed 192 sites, bin 10, the fastest category of sites, and we got 87 to 90% bootstrap support, depending on whether you used parsimony or likelihood, for the human chimp tree. So it didn't have a tree in the beginning. It removed what it considered to be the most weird sites, most unusual, this proxy for rate, and we get reasonably good support for human and chimp, and no support for the other. And so I think this. That's about as far as this method goes in terms of what it does right now. I think there may be things in the future, but I thought it might be interesting because of the audience that's here to say what kind of questions arise at this point, like at least in my head. So first of all, are all tree-based approaches inherently systematically biased? Are all tree-based methods for trying to evaluate the rates of evolution of sites, are they all inherently biased? I don't know the answer to that. Is there a tree-based method? People have suggested to me, why use a single tree? Maybe use the entire Bayesian, output from a Bayesian analysis, so multiple trees. I think that might be a good help. I don't know the answer. We haven't done any work on that, so if people feel it's sufficiently interesting. Is there a maximum likelihood alternative to comparing pairwise comparisons of sites? I've had just some discussions with people on that. Is the discrete approximation to the gamma distribution adequate? Is the gamma distribution correct? Is it adequate? People love the gamma distribution. It really improves the likelihood of your data. But are there other distributions out there that might do a better job? What about the beta distribution? I have 2 slides coming in a second that might talk a little bit more about this. What do bootstrap support values mean when the data has been manipulated in advance? So again, that's something I think we've talked about earlier. Are branch lengths still useful? When you've lopped off a number of sites you don't like, what does the branch length now mean? When do you stop removing characters? This is a question we have no really good answer for. I think Lars has a method for stopping, so we're going to have a chat about that. But at what point do you stop? Does it stop becoming ad hoc? And do you have an objective scientific way of removing characters? And of course, what's the relationship between rate and structure? Could you use this kind of method to tell you something about protein structure and protein evolution in that way? So this is a slide that I showed you earlier, showing that you can pick out pretty subtle patterns in the data, differences in rate. I think this picked out the fact that there were 4 different rates, 4 different absolute rates built into the way the data was simulated. Well, this is a real data set. This is the primate data set. And you've got a lot of constant sites here. Is it the primate data set? Yes, it is. A lot of constant sites. Nothing here. And lots of fast sites. So I don't know what that means either. Okay, if you want the software, it's here. It's on this website. It's written in Python, so it should be multi-platform. As I said in the beginning, one of the things that I wanted to talk about here were non-tree-based methods. And my lab is involved in, if you like, a program of research where we're trying to stop thinking about trees, because there are other ways of looking at data, I think. And on one hand, we may look at alignments, but on the other hand, we may think about, sort of, if you like, bigger issues and what this means. And so I wanted to devote a little bit of time to talking about this and talking about I guess I think it's probably the biggest issue we have in evolutionary biology, but that's my own personal opinion, and that is how we describe life on the planet. What is the top theory, the very high level one, the one that says this is how evolving entities on the planet evolve. And I've been doing this work with Eric Baptiste in Paris, and David Epizani in Maynooth, and Mary O'Connell in Dublin City University. And we've got a paper, and if people want to have a look at it, I think it's going to come out in Biology Direct. And so there's a little bit of history here and a little bit of philosophy, and I hope it doesn't bore you, because I know that it's very, very different. But the reason I put up slides like this is from a paper by Mark Reagan, where he dug out all of the ideas about the evolution of life on the planet. And the Scala Naturae is probably something that you're all pretty familiar with, but this is an hypothesis for how life on the planet is structured, not just life, but also rocks and water and stuff, and gods. And so this persisted for some hundreds of years. But there were also tree-like structures that people were putting together to describe life. I quite like this one. It's sort of the asparagus bunch of life on the planet, which existed as well. But also net-like structures, and you're probably all familiar with these. These are quite reticulating here. These are plants, and these are vegetables here, and this is the phylogenetic history of dogs. And people have been putting these together as a way of looking at life on the planet. But I think everybody got very, very influenced by the way that Darwin structured his thoughts in this area. And he said that he thought effectively that the metaphor of a tree was the most important. I want to dwell on this slide just for a second. You've probably all read this, but effectively it says the affinities of all beings of the same class have sometimes been represented by, I think that's important, a great tree. In other words, that this is central. And from the tree, all of the things sort of come out. That there is a great process in the middle. And it's a constantly diverging process. It says it here later on. I believe this simile largely speaks the truth. The green budding twigs may represent existing species, and those produced during former years may represent the long succession of extinct species. The limbs, divided into great branches, and these into lesser and lesser branches, were themselves once, when the tree was young, budding twigs. And this connection of the former and present buds by ramifying branches may well represent the classification of all extinct and living species in groups subordinate to groups. So groups subordinate to groups. This isn't just saying that trees are nice pictures, but it's actually saying that there's a central process that's driving evolution. That there is a central tree, it's constantly diverging. And this idea, of course, is something that people have really, really liked. Now, it's been manipulated, it's been changed over the last 150 years. And in order to actually talk about what I want to talk about here, I think first of all we have to define what the tree of life is, or what people think the tree of life is. Carl Woese was tremendously successful in his idea that there was this great molecular chronometer, the 16S or 18S ribosomal RNA. It had these lovely properties. All cellular life have ribosomes, so therefore we could use this to compare E. coli and elephants. And that was really good. But if you read the papers, Woese didn't really spend a lot of time speculating about a tree of life. It was the case that we may have a tool in order to infer a tree of life. Ribosomal RNAs best fit this, so let's go and do this. And it's been hugely successful. The most sequenced gene on the planet by directed sequencing is this gene. I'll come back to the most sequenced gene on the planet later. Concatenated or widely distributed genes, a highly controversial paper published about 5 years ago out of Per Bork's group, Ciccarelli et al. Concatenated 31 fairly universally distributed genes and inferred a phylogenetic tree and said this is progress towards a tree of life. It's been heavily criticized. Tal Degan and Bill Martin have published that this is just a tree of 1%, so how can this be a tree of life? And more recently, Pere Buijbo and Eugene Koonin have been looking at things like the forest of life and the statistical tree of life, a sort of more fuzzy or furry sort of version of a tree. And of course, we've got people trying to integrate entire genomes by making phylogenetic supertrees. There's a lot that could be said about this slide that could probably take us a day to get through. But the same idea exists here, that there's a great central tree onto which we map things. And more than that there's a great central process. There are problems with it. And I thought this week has been particularly instructive in seeing how many people are working on this and from how many different angles. There are problems. First of all, what's the most abundant gene on the planet?
Control region of the mitochondria. Control region of the mitochondria.
This is gotten by metagenome sequencing, by going out, sequencing things in the ocean, on soil. It's a transposase. Transposases are the most abundant genes on the planet. The most highly sequenced now, probably, because we're sequencing so many metagenomes. But transposases, wouldn't we have a very different picture of evolution on the planet if the first genes that we ever sequenced were the most abundant? Not the one that's found in cellular life. Problems with the tree of life. Conjugation by plasmids is not predicted by the tree of life hypothesis. Transformation by phages is not predicted by the tree of life. Natural competence of organisms such as Neisseria is not predicted by the tree of life. The secretion of outer membrane vesicles, you may not know about this one, but certain bacteria, when you stress them, they bleb off little bits of membrane and they set them free. It's the equivalent of you, as Bill Martin says, of you cutting off your own toes and handing them to somebody else, and they integrating them into their body. Gene transfer agents, which are kind of an unusual virus-like thing. Nanotubes, 2011, about a month ago. Publication showing that cells, if they just touch off each other, they set up nanotubes which passes DNA from one cell to another. Not predicted by the tree of life. Restriction modification systems, defense, not predicted by the tree of life. Why would you want a defense system if everything grows apart, if everything evolves divergently? The complexity hypothesis, the Rivera and Lake complexity hypothesis, which says that the rate of horizontal gene transfer of informational genes is much, much lower than the rate of horizontal gene transfer of operational genes. This has been updated recently by a very, very, very good paper from Tal Pupko's group. And in this paper, they show that, in fact, one of the main features is that highly connected proteins rarely undergo horizontal gene transfer. It's the connectivity in a protein-protein interaction network that determines how much you're going to be involved in horizontal gene transfer, not predicted by the tree of life. The ring of life hypothesis from Jim Lake, not predicted by the tree of life. The hydrogen hypothesis for the origin of the eukaryote, not predicted by the tree of life. Dennis Searcy's sulfur syntrophy hypothesis, not predicted by the tree of life. Endosymbiosis, not predicted by the tree of life. Hybrids, and we've heard a lot this week about hybrids, not predicted by the tree of life. Clubs and coalitions of microorganisms, sharing, metabolisms, not predicted by the tree of life. But if ever there was a diagram that actually presented the tree of life hypothesis with its biggest problem, it came out of David Ustery's lab last year. It's this one, it's figure 4. I can give you the reference. Here. Comparison of 61 sequenced E. coli genomes, published in Microbial Ecology. What you have here on this axis is 1 to 63 genomes. They're listed here. And what you have here are the numbers of genes that are, in red, universally distributed across all of these sequenced genomes. And in blue, the total number of gene families we find. So you sequence the first genome, you get 4,000 and something genes. The second, the third, and suddenly the number of genes that are universally distributed across all of just E. coli is down to about 500. The total number of genes we find in E. coli so far, and that doesn't look like it's about to plateau yet, is about 15,000 genes. Okay? This is not predicted by the tree of life hypothesis. So there are issues in it. And the way in which these data have been dealt with has always been to say, well, we must change the tree of life hypothesis a bit. We must hang cobwebs off of it, or we sort of bend these branches and stick them together. We must invoke more ad hoc hypotheses, and more and more and more. I have no idea how many ad hoc hypotheses you have to invoke to say this is tree-like evolution. 15,000 genes, not one genome is more than about 5,500 genes. And when you just look across this one species of prokaryote, the number of constant genes that are found in all the genomes, is a fraction of the total number. Not predicted by the tree of life hypothesis. So we approached this from an axiomatic perspective. We said, if you're going to go and park tree of life notions for a minute, and you're going to think about the evolution of life on the planet, the top level, where do you start from? Well, these are two, I guess, uncontroversial starting points. Based on what we know about life on the planet, first of all, that all evolving entities should be included in a model that aims at providing the largest evolutionary picture. So we can't leave anything out. That's maybe a good starting point. Let's include everything. The tree of life doesn't include viruses, and it doesn't include plasmids, it doesn't include phage. So we prefer not to leave anything out. Genes, they're inherited both horizontally and vertically. That's fairly uncontroversial. I think it's easy to show that that is the truth. So our hypotheses can't violate that. Our idea can't violate that. But how to do this without thinking, first of all, about trees, number one. And number two, we can make trees, we can make phylogenetic trees, and indeed we can see compatibility or similarity between different trees. We can see tree likeness, but perhaps it doesn't describe everything. And so where we looked was in economics. And this is Paul Anthony Samuelson, who won the Nobel Prize in 1970, the Nobel Prize in Economics in 1970. He died about 2 years ago. And even though about 400 years ago, John Locke discussed goods and government, I think Samuelson is the person who's being credited for describing the kinds of goods that exist. And he wrote this in 1954. He said, I explicitly assume two categories of goods: ordinary private consumption goods, which can be parceled out among different individuals, and collective consumption goods, which all enjoy in common, in the sense that each individual's consumption of such a good leads to no subtraction from any other individual's consumption of that good. And so what we're doing is we're asking the question, do genes have the property of being like economic goods? Moving on from 1954, it's generally accepted now that there are four kinds of goods. And it depends on whether they're excludable or whether they're rival. Okay? So if a good is excludable and rival, it's said to be a private good. So if I've got a cup of coffee, okay, I can exclude you from having that cup of coffee, and my usage of the cup of coffee means that it's not available anymore even. So it's a private good. And so on. Common goods are non-excludable but rival. Club goods are said to be excludable and non-rival, and public goods are non-excludable and non-rival. So I want to talk about these a little bit. So as I said, the cup of coffee could be said to be a private good. Golf clubs can be considered to be club goods because they have the property of being excludable but non-rival. If you're in the club, you can use the club, but you can be excluded from the club. Private street lights are common goods. If I own a shop and I'm trying to encourage customers in, I can put a little light outside the door, and everybody walks along and says, this is nice and bright, I'm going to go in and buy something. So it has the property of being non-excludable. Anybody can use that light, but it is rival in the sense that the use, not everybody has access to it. The usage of this light is considered rival. Air is a public good. Public goods tend to be a little bit intangible, and they also tend to be a little theoretical, in the sense that air is a public good. My usage of air is not preventing any of you from using it, and even if I wanted to, I can't exclude all the living things on the planet from having that air, so it's a public good. Knowledge is considered to be a public good quite often. Files on the internet are quite often considered to be a public good, or in many cases they can be. In other words, when a file gets on the internet, it's very, very difficult to stop people from accessing it if it becomes freely available, and also the usage of that file, because it's copied, means that when one person has it, it's not being used up from somebody else. Somebody else can have it as well. So these are goods. So are homologous gene families goods? Can we explore this idea that homologous gene families are goods, and indeed that theoretically they are public goods, but maybe practically they're club goods or private goods or common goods? And so what we're writing up is what we call the public goods hypothesis, and it's described, I guess, in this slide. The genes are potentially public goods. This is because, first of all living things can use them. They're made up of nucleotides. There aren't different kinds of ways of encoding genes. There are the 4 nucleotides, or I guess maybe the 5 nucleotides. So they're free to be used by anybody. There's a universal genetic code, except for, of course, the variants, and they fit into it as well, because you can privatize your genetic code if you like. Things like mycoplasma and spiroplasma use a variant genetic code. But in general, these are just at the tips of the trees. I guess mitochondria are there as well. But mitochondria don't undergo gene exchange. Not so much. And gene mobility, a mechanism of moving the goods around. Does that exist? And the answer is yes, it does. So we've got natural competence, phage, gene transfer agents, and so on. And these mobile elements seem to be the most abundant kinds of genetic element on the planet. Genes are acquired by a genome and are passed vertically by replication and division. So this might lead to some kind of tree-like pattern. When you compare one gene tree to another, you might say there's some agreement between these. Of course there is. They're in the same genome. They're passed down vertically. We know this happens. So we expect that even if genes are public goods, theoretically, that they may you may find, when you reconstruct their histories, that genes traveled together. Just as many people got here on the same train, we travel together, but that doesn't mean that you're always going in the same direction. In practical terms, loci may be privatized or brought into a club. And I think things like we see quite often the symbionts of aphids are inherited exclusively by the offspring of those aphids. And there's a kind of genetic club there, which is not predicted by the tree of life hypothesis. And it satisfies the two central axioms. We don't leave out plasmids, we don't leave out phage, we don't leave out viruses. Mobile genetic elements have a place. Horizontal gene transfer is central, and a tree-like thing may emerge because of gene duplication, genome duplication, and cell division. A tree-like thing, but I don't expect it to be very, very strong, and that's what we see. Okay, can genes be excludable? Alternative genetic codes might do this. Very, very high connectivity in a protein-protein interaction network might contribute to gene excludability. So I said about public goods, they're slightly intangible. I said air was a public good, but not if we're all underwater and I've got a bottle of air, then it's mine. It's my private good. So there's a context dependency on whether something might potentially be a public good, theoretically, and whether it is a public good, actually. And things like this may modify its status. Could genes be rivalrous? And we're kind of trying to explore this a little bit. Plasmid incompatibility. So for some large plasmids, INC-H plasmids, you can't have two versions in the same cell. They will segregate and they will go into different daughter cells. And we can see competition potentially between alleles as well. So rivalry between alleles, which might lead to them being private goods or otherwise. The tree of life implies only private goods. It implies a group within a group within a group. Privatization. Okay? The public goods hypothesis says there can be any of these. It's a much more pluralistic approach. It's a much more relaxed way of looking at it. If we look at a table and we look at evolutionary things, the evolutionary entities, do we expect to see a single or central tree? According to the public goods hypothesis, no. According to the tree of life hypothesis, yes. Alternative genetic codes, yeah, that's fine. And the tree of life says yes. In fact, I would almost expect that if life evolved in a sort of tree-like way, that we might see things, alternative genetic codes, more extensively. So mycoplasma and spiroplasma are two sort of, if you like, tips, are tip nodes on the prokaryotic phylogeny. But we don't see half the prokaryotes having one genetic code and half the prokaryotes having an alternative. It seems that you might have expected this if there was no horizontal gene transfer, if there was a central tree. Do we expect to see very different trees? Public goods says yes, tree of life says no. Is the complexity hypothesis expected? Well, it's fine under this scenario, but not under this. Plasmids, viruses, gene transfer agents, outer membrane vesicles, natural competence, nanotubes, CRISPRs. We had a very nice talk this week about CRISPRs. Why would you want a defense if everything evolved in a tree-like fashion? Endosymbiosis, horizontal gene transfer, coalitions, restriction modification systems, hybrids, and ring of life. So, I think I may have said most of this already. The public goods hypothesis predicts weak local trees of cells or viruses or plasmids. Any tree likeness is a consequence of public goods. It's a consequence, quite often, of horizontal gene transfer, followed by genome duplication and cell division, genes passing along together for quite some time. Sometimes only for a short time, sometimes for very, very long. And nearly universal trees from Eugene Koonen's lab, those are genes that, because of natural selection, have stayed together for a long time. It predicts trees of genes. It does not predict a great unifying tree. And I guess more importantly, it doesn't predict a great unifying process. That the central process is not a tree. The most important process is something that's a little bit more all-encompassing. It's horizontal gene transfer followed by vertical evolution for whatever length of time, gene division, genome division, and horizontal gene transfer again. And most explicitly, because this is what we're trying to achieve, it does not start with the assumption of a tree. And that means that there's no conflation of explanandum and explanans. And this is the problem with many efforts to understand the tree of life, is that you start with the assumption that there is a tree, and then find something tree-like which proves that there is a tree. the exponandum and the exponans are conflated, and that's not something that's a very, very good idea. So where stands the tree of life at that stage, or at this stage? At least for me. I think it's a regionalized hypothesis. If you go back in time, Newtonian and Hamiltonian physics 150 years ago was physics. It was supplanted by a much sort of broader idea called quantum physics. It's not that Newtonian and Hamiltonian physics doesn't work. You know, if I push down one side of a seesaw, the other side goes up. It's just that it's inadequate to explain everything. Euclidean geometry versus Riemannian geometry. Again, the same idea. Three-dimensional space is very, very nice. It's very comforting. I can see it. However, it doesn't include everything. And I think while Euclidean geometry is perfectly fine still, I believe that it's insufficient to cover everything. And in terms of tree of life, I think we can look at it somewhat like this. And this is an incomplete diagram, but it sort of gets across the idea. The within public goods hypothesis, you have a variety of other hypotheses. These are the tree-like ones here. Things like the ribosomal RNA or single gene tree of life ideas are contained within multi-gene tree of life ideas. And the statistical tree of life that Eugene Koonin has been writing about for some time is sort of, if you like, a superset of this idea as well. Or in other words, that this has been regionalized under this idea, and this idea has been regionalized under this idea. However, these do not account for things like mobile genetic elements because they're tree-like. Complexity hypothesis, the ring of life, endosymbiosis, and rather than having a diagram that was just too complicated, I've stopped there. But the public goods hypothesis sees a central function for horizontal gene transfer between membrane-enclosed lineages. It sees a place for all evolving entities. It's the last slide anyway. It sees a place for all evolving entities. It has a process that doesn't have a tree at its heart, but it sees a tree as a consequence of these various processes. And I'm going to finish there. Carla Cummins developed the Tiger algorithm for looking at alignments. In terms of public goods hypothesis, I've been working with Eric Baptiste, who's a philosopher and scientist in Paris, Davide Pisani and Mary O'Connell, who both working with me in Ireland. Funding was from Science Foundation Ireland. Mark Wilkinson, I'd like to thank him for several pages of very helpful comments. And the public goods hypothesis is, I think, to be published in Biology Direct, and it's already been reviewed by Ford and Eugene and John Dupre and Davide Vecchi and Greg Morgan. I'd like to say thanks again to the organisers and thank you for listening.
Any questions?
How dare you?
The lady was first.
Yeah.
I really like to talk just towards the end with your non-tree thinking. So it seems to me that the advantage of having a sort of tree-like idea is that you have some predictive power, right?
It's a heuristic. So heuristic is a word that gets used differently in computer science than its original incarnation. A heuristic is a rule of thumb. Is it useful for something? It's useful for taxonomy, I think, yeah?
But then it seems to me that the public goods hypothesis doesn't really have any predictive power.
Oh, it does.
Can you explain it?
I'm going to predict that we will find more origins of replication. Okay, I think we're going to find more viruses. I think we're going to find more coalitions. I think we're going to find more symbioses. I know of a paper that's under review right now, or sorry, under submission right now. I don't know if it got chucked out, where they've discovered another ring of life, so another genome fusion. Okay, so I haven't seen the data on that, but I've been told that it exists. So yes, it does have predictive power in the sense that that to a certain extent, and only a little bit, I think, really, tree thinking stops us from thinking laterally. And not in this group, I think, because this is a very lateral group. But in general, we expect divergence, and in fact, I think, we see convergence, and we start with the tree when in fact we should end with the tree.
And I guess that was kind of relating to a second point. Was do you think this idea explains why there's so little disparity versus diversity, in terms of if you look at organisms at the moment, they're not that as disparate as you would expect. The diversity seems to be tinkering around on the edges.
Okay. I work with prokaryotes, and they're way more disparate than I expect them to be. I also didn't mention much about eukaryotes and meiosis. So meiosis, it fits into this, but I think it, largely horizontal gene transfer has been, gene exchange has been fairly much privatized. But of course, we've seen recently Wolbachia, a prokaryote, has its host's DNA, great big chunk of it in its own genome. So it happens to a certain extent. It's still there. Again, Wolbachia having host DNA, insect DNA, is not predicted by the tree of life hypothesis. Yeah.
News is not predicted by the tree of life hypothesis.
Sorry?
Fox News is not predicted by the tree of life hypothesis. I mean, it's a bit ridiculous just making a list of all the things it doesn't predict. It's not meant to predict everything in the universe. It's meant to say something about the general processes that occur. And generally, replication is a pretty major process when it comes to making organisms.
Yeah, that's if you look at it from the perspective of replication, but transposition is a very, very major one. And in fact, in terms of magnitude, it's bigger. And in terms of horizontal gene or if you in terms of tree of life, you get a tree of 1% of any genome.
Explain why the magnitude's bigger?
Why it's magnitude?
Yeah, where's that result come from?
It comes from the fact that the number of genes that you can put together that are compatible with each other is much, much smaller than the number of genes that are out there. Conflict is higher than compatibility.
Right. And you said before that transposons and their action are more prevalent?
Transposases are more numerous. Sorry? Transposases are more numerous than ribosomal RNAs.
I think we should let you discuss this in private because there were some other questions, if this is okay with you, Alex. So there's two more
kind
questions.
of like slightly to what Alex was saying anyway. I guess predictive power also involves having things that you don't predict. So
Right.
what would cause you to throw away your hypothesis? What single observation would say this isn't true? Because I can't see one.
Yeah, so
You
this
do
is...
admit that you're doing philosophy. So
Sorry?
I guess
Yeah,
you're
so
doing
this
philosophy,
is
so you're
the
allowed-
idea of whether this is a model or an hypothesis. So I think it's much more like a model than it is like an hypothesis. I think it's going to be, yeah, I think that's a very good question. Yeah.
Lars, please.
Your some support from the fact that the different genes, different data sets all suggest that there are that different genes have different evolutionary histories, and because there is this lack of consistency across those, then there can't really be a tree of life. But that I think I would be cautious if I were you, because the vast majority of those data sets that I have looked at to date using some methods that we have developed to determine whether there's evidence of model misspecification or not, suggest that there's rampant amount of model misspecification. And before we have actually managed to account appropriately for that using much more general marker models, for example, I don't really think it's worthwhile debunking a tree of life.
Not debunking it. Not debunking it. It said it's regionalized.
Or, well, even that.
Just going to go back to this. Sorry.
So you took an example. Yeah, well, that one I like. But you also referred, for example, back to Ciccarelli et al.'s paper.
Mm-hmm.
Now, Ciccarelli et al.'s paper, if you actually read that very, very carefully, what they did was that they aligned first all the eukaryote genes, then they aligned all the bacterial genes, and then finally the archaea genes. And then they aligned those three alignments.
Yeah.
Now, unfortunately, what they end up doing there is that they created an alignment that has all the hallmarks of being an alignment that is not appropriate to derive. If you look at the composition across those in terms of amino acids, you find massive differences in the amino acid composition across those. You find evidence of covariant evolution, very, very much. Different domains have different sites that are invariable. So the distribution of those invariable sites are completely domain specific.
The Pear's group have just had a paper accepted in PLOS One, I think. I'm not certain, one of the PLOS journals, where they show that none of the genes that they used are free of horizontal gene transfer effects.
Lovely.
That means that we're down to 0 genes that we can find that have not undergone horizontal gene transfer.
Yeah.
We're down to 0. So for me, there's an issue here. If it also comes down to comes down to this slide. You almost have to read the paper to find out what the author means when they say tree of life. That's an issue right now. It's a definition of what it is we're actually talking about. For me, it's a central process. It says that the process centrally is tree-like, diverging, and so on. And the data doesn't stand up to it. You know, and it ignores plasmids, it ignores viruses, it ignores phage, it ignores gene transfer agents. And so it has problems with inadequacy, and it has problems with the data doesn't support it.
But I mean, that's a total straw man, because the tree of life, everybody knows there's horizontal gene transfer. Bloody hell.
Okay.
But what's your proposition? No, I think it's good. Alexi, what's the proposition? What's the proposition?
You should have a model.
Okay,
I mean,
can we just 1 second now? So if I drop this on the floor, it falls. Okay, Newtonian physics. I'm not saying the Newtonian physics doesn't work. Sometimes I'm saying it doesn't work. No one's going to say that's the only physics.
Sure. That's why it's a straw man.
It's not a straw man. It's not a straw man. I'm saying that there is an inadequate way of describing life on the planet.
Okay. Now my problem is we are 5 minutes over time.
Okay, sorry.
I can see there are many questions. There are many open discussion points that you could discuss. So we can either go on for 5 more minutes, or if everybody is happy with that idea, we could go for a democratic idea, which means we just raise our hands and then we go for that, or we stop now and we just defer all the discussions we might have to after this talk or to the Hogwarts party later tonight, because it's a kind of philosophical